Thank you very much for the very kind introduction. It's such a pleasure to be here today. I'd like to talk about the genetic determinants of one of the hallmarks of aging, which is telomere attrition. So I understand that a lot of you here may not study telomere, but I hope that my presentation here inspire or sort of illustrate, especially to the younger investigators, how we look at one topic and we move from studying the genes, from the simple SNP associations, finding the truly functional gene, and all the way to understanding its function and its impact on population risk and the mechanisms linking it to the actual important outcomes like mortality. Yeah, okay. So telomeres are repetitive sequences of DNA sequences, and they are associated with the sheltering proteins, and they cap the ends of the chromosomes. So they protect the individual's chromosomes from degradation and from the interchromosomal fusion events. However, because of the inability of the DNA replication machinery to fully replicate the lagging 3' DNA strand, telomeres naturally shorten with each cell replication. So, we usually start with about 10 kilobase in humans, and then with each cell cycle, it shortens naturally. And so telomere length determines the lifespan of a cell, because when it's shortened within a critical length, that's when apoptosis is induced and the cell dies. And that's why telomere attrition has been recognized as one of the hallmarks of aging. So as mentioned, aging, despite all the lifestyle factors that we know can affect aging, is still determined genetically to a certain extent. And for telomere length, it is actually a highly polygenic trait. But people have estimated that it's also highly heritable, up to about 40%. And most variations in adult telomere is actually set at birth. So Brian mentioned about, the man being genetically wired to have shorter lifespan, with events that begin in the womb. And true enough, also, we have found that most variations in the leukocyte telomere is set at birth. Baby girls at birth have longer telomeres than baby boys. You might think, okay, for once, the gender inequality is in favor of the woman. So we also know that there are potential ethnic differences. For example, Europeans have shorter telomere length compared to the sub-Saharan Africans. And you might say, hey, that's not quite fair, because life can be harsher in the African continent with higher parasitic and infectious loads. And that's precisely why evolutionary pressure actually confers longer telomere length to the sub-Saharan African population, to help them, actually, to give them that advantage for the harsher environment that they face. So we also know that shorter telomere length is actually linked to higher risk of chronic diseases. Lots of associative studies showing that shorter telomere length, higher risk of mortality from heart disease, neurological degenerative diseases. But, paradoxically, the higher telomere length has also been associated with higher cancer risk, because we know that the enzyme telomerase from cancer cells can actually also lengthen telomeres. So it is not like the longer the better. There is a sweet spot, because there are adverse risk events associated with short or long telomeres. And so we know that telomere length is therefore complexly related to aging and disease risk. And so we asked ourselves the questions, how can we study the genetic determinants of telomere length, and how will this knowledge help us understand the link between telomere length and age-related phenotypes and disease risk? Of course, we have a lot of European studies that have raised the head and gone and identified at least 8 genetic loci or more than that. But these genetic loci put together only explain about 2% of the phenotypic variation. So a lot more work remains to be done, and we believe that we need actually to do not just another population, but another population different from the European population. And so we turn to the Singapore Chinese Health Study that I'm a PI of, and this is a population mainly of Southern Han Chinese ethnic group. So these are the Chinese that have migrated to Singapore from China in the 1930s, coming to Singapore to build the nation. So as you know, only the Malays are really indigenous people here. The Chinese and Indians are actually migrants, and we are descendants of the migrants. So we have a discovery cohort of about 16,000 people, and within that we kept aside about 6,000 for our discovery cohort and our replication cohort. So this is Singapore-Chinese self-study, started in the 1990s, some 30 years ago. So for young people here, think of your grandparents, and those will be the typical cohort participants that we recruited. Repeated follow-up, over about coming to 30 years now. So the work that I'm presenting had at least close to 25 years of follow-up. And we actually followed them up for aging outcomes just 10 years ago, when the participants were about average age of 73. And we also had regular linkage for cancer and death outcomes. Importantly, in between 1999 to 2004, we managed to go to 32,000 of these participants and got them to donate blood or Barker cells for DNA. And we have finished GWAS genotyping with the Illumina chip for about 23,000 subjects with high quality data. We also, at the same time, measure the telomere length in their leukocytes using qPCR method. So at that time, for this population, the mean age was 62, the range was 45 to 85. So we did get data of midlife telomere length and also the accompanying GWAS in these individuals. And that enabled our first study looking at the association between SNPs and telomere length. And the first thing we did was, of course, we confirmed, want to see whether we could confirm what the Europeans had published, and true enough, true enough, we're able to confirm four previously published, genetic loci, the TERT, TERT-C, and the NAFL, three SNPs, three loci. And then there were two loci that were previously shown in European populations, but we were able to find new SNP variants. And we finally also more importantly, discovered 5 novel variants at POT-T, TERF1, ATM, TINF2, and the PARP-P1 loci. So we had new variants not previously reported in the European study in association with telomere length. So, of course, we wanted to make sure that the novel variants are actually, true variants. And so what we did was to look at so we looked at our top variant, which was novel, and this was actually the TINF2 variant. And this had a p value for the association of a very high significance of 10 to the power of -42. It's completely novel. And when we went to the 1,000 Genome Project, and we looked at the minor allele frequency, we see that actually this was completely monomorphic in the Europeans and the South Asian cohort. So this allele variant is only found in East Asian, and would have been completely missed if we didn't study East Asian cohort. We also found this for the TINF2 SNP, and again, another very novel variant genetic loci that we found. And again, we found that this SNP is also cannot be found among the Europeans and the South Asian population in the 1,000 Genome Project. And the recent, much later, the UK Biobank reported on the TIN2 SNP, but that's because they worked on 500,000 European subjects. And we were able to see that in just 23,000 of our population in our cohort. And so this shows you the importance of studying, doing this type of studies in different populations, because we do find novel genes due to the fact that some of them may actually be monomorphic in other populations. And even for the ones that were for the genetic loci already presented in the European populations, we found new genetic variants. So for the OBFC1 gene and the RTEL1 gene, the loci that we found were different, and they also had differences in the linkage disequilibrium. And this would therefore potentially actually point us to different functional genes. And so we asked ourselves, true, it's nice to find novel genes, publish and report them, but are these real? So we wanted to know whether the genes that we identified could actually really be involved in telomere homeostasis and DNA regulation. So for that, we examined about 2,000 SNPs that were in linkage disequilibrium with the 16 lead GWAS SNPs in our study, and we mapped them to 268 genes located within 200 kilobase kilobase pairs of these lead signals using the gene ontology and the canonical pathway analysis. And indeed, we found that most of these genes were really enriched in pathways associated with telomere and DNA repair, such as the genes that were, such as the telomere maintenance pathway, involved in telomere lengthening, telomerase activity, base excision repair, DNA double-strand break pairs. And then we were really pleased that there were genes definitely coding for proteins involved in the sheltering complex and DNA repair, and also in the telomerase assembly. So these NIPs were able to link us to genes that were likely to be functionally involved in DNA homeostasis. And then we wanted to link the index SNP to the functional gene. To say, it's like you found somebody and you want to use this person as a clue to link you to the real person involved in the mechanism. And the straightforward case is actually the novel PARP-P1 variant. And this is easy because this one is actually in a coding variant, so it's likely to be functional. And we look at the CAT score, which tells us the likelihood of deleteriousness of the variant. It is above the cutoff of what is usually accepted, is above 30. And more importantly, we know that the PARP-P1 is involved in telomere homeostasis because it modifies, it binds to and modifies another protein, the TRF1, which can go on to stabilize the DNA for accurate DNA replication. So this gene is very likely to be real and functional. But most of the other genetic loci that are novel that we found were in the intronic and intergenic regions, and therefore it's harder to see how they are related. So we have to work a little harder. So for that, we went to map the 2000 SNPs, and then we linked them to we tested whether the GWAS identified loci could regulate gene expression in blood immune cells, since we studied leukocyte telomere length, and we used these various QTL data sets shown here. And we also performed localization analysis to see whether the QTL signals and our GWAS signals could actually point to the same causal variants. And we are really pleased that indeed we did find at least 5 genes here, 5 genes here, which are top genes with high probability of sharing causal regulatory effects. And then more importantly, we found that actually the rare variants, those who are less than 1% MAF, actually may have stronger effects than the common variants. For example, one of the rare variants is the POP-T1 gene that was imputed using our GWAS data, and we actually found that the minor allele actually had 5 times greater effect than the common allele. And that again tells us that if we study rare variants, it can actually improve our knowledge in telomere control. Okay, so, but what's the point of having all these genetic loci? We really wanted to know whether they are really impacting population risk for mortality and disease. So for that, we created a gene risk score from for telomere length from the 21 low frequency and common variants reported to be associated with telomere length. So we created this genetic score, and we studied cancer risk. And we found that indeed it was significantly associated with the risk of colon cancer as well as with lung cancer. And you can look at the results here with high statistical significance. And then we wanted to see whether the genetic risk scores association could really be mediated through the midlife telomere length. And indeed, we were very pleased to find that for the lung cancer, as high as 25% of the genetic effect on lung cancer risk was mediated through the midlife telomere length. For colon cancer, it was lower, but still statistically significant. At least 5% of the genetic effect was mediated through the actual midlife telomere length. So for mortality, we had 16 variants, so we needed to be very stringent to correct for multitasking. And we found that indeed there was one genetic variant that was highly associated with respiratory mortalities. And this was associated with telomere length shortening, and our co-localization analysis potentially pointed this gene to immune cell gene expression. And so again, we wanted to see whether the effect of this gene on mortality could be mediated through the leukocyte telomere length, and it was a significant, though modest, 3%. All right, so then we looked at other aging outcomes, including depression, gait speed, cognitive function, and we found that the genetic risk score and telomere lengths were all associated with hand grip strength at the age of about 70, so at late life hand grip strength. And again, we found that the association of the gene with the hand grip strength was also mediated through midlife telomere length up to about 33%. So, really pointing out to us that telomere length affected grip strength, which is actually a marker of sarcopenia. So much of the heritability, however, remains to be uncovered. You can see that even with all the genes from our cohort and the European cohort, we still could only explain up to just about 5% of the genetic variability. So how do we find the rest of the genetic variants? So we have resorted to whole exome sequencing. This is work in progress. And so I wouldn't say much here also because I don't have enough time. So we have done a whole exome sequencing of about 8,000 samples. We sampled from the two extreme ends of the short and the high telomere length, and also a portion from those with average telomere length. We have finished sequencing. We're now analyzing. We finished analyzing the data, and we wanted to look for genetic variants that could be linked to potentially functional mutations. So I think this should be my last slide because I would just reveal that we have found a novel association at the BLM locus that codes for the Bloom's helicase that reaches GWAS significance of 10 to the power of -7. And so this was actually reported in the UK Biobank, but did not reach statistical significance there. And I'm sure you guessed the answer. Again, it's because it's almost monomorphic in those populations, but we do have sufficient minor allele frequency here. So our work therefore shows that even if other people have done studies in other populations, there is a need for population-specific studies because that could actually reveal a lot more about the underlying genes and therefore potential genetic targets for risk stratification and also potential protein targets for modification as therapeutic agents in the future. So right now we are looking at 2 time point telomere attrition. We are looking at discovering more genetic as well as the actual protein, determinants, and this will be work which I hope to share in the future. I will not talk about the SG70 because we've just finished recruitment, so work is in progress, hoping in the future, very soon I'll have more to share. I will stop here because, but it's so important. The work is done really by my collaborators, especially Raj from GIS, Alfata, who is in the audience. We are now looking at the function of this Bloom variant that we found using yeast-based functional studies. Sorry, I think I exceeded the time, but thank you very much for your attention.
Thank you, Professor Koh, for the great talk. We have time for a few questions.
Yeah, we do. I'm so sorry. 40.
Yes, 2 minutes.
Yes.
Yes.
Kind of a technical question, but when you look in UK Biobank, there are Asian people in that population.
Mm.
Do they account for like the TNF2 alleles, or the other one that you said almost reached significance? Is that enriched in there?
Yeah. Yeah, I know. So the UK Biobank is still predominantly Europeans. So they do have, and that could be the reason why, as I mentioned, that the TIM-F1, which they later on also reported, finally, when the sample size was 500,000. And we think it's because they finally saw that the flicker of light from the Asian population, because it's monomorphic in the 1,000 Genome Project. Yeah.
Okay, thank you very much.
All right, thank you.